Question 21. Contributions of Louis Pasteur. Answer: Louis Pasteur, 1822 to 1895, was a French scientist who made significant contributions to various fields of science, medicine, and industry. His work had a profound impact on our understanding of microbiology, disease prevention, and vaccination. Here are some of his notable contributions. Contributions of Louis Pasteur include: germ theory of disease. Perhaps Pasteur's most significant contribution was the development of the germ theory of disease. He demonstrated that many diseases are caused by microorganisms, germs, such as bacteria, rather than by spontaneous generation or other mystical causes. This theory revolutionized medicine and led to the understanding of the role of microbes in causing and spreading diseases. Pasteurization. Pasteurization is a process of heating liquids, especially milk and other beverages, to a specific temperature to kill harmful bacteria and pathogens while preserving the flavor and nutritional value. Pasteur's work in this area has been instrumental in improving food safety and preventing the spread of diseases through contaminated food and beverages. Vaccination. Pasteur is often referred to as the father of immunology due to his pioneering work in developing vaccines. He created the first successful vaccines for several diseases, including anthrax and rabies. His techniques for weakening or attenuating the virulence of pathogens to create vaccines laid the foundation for modern vaccination practices. Question 22. Autoclave. Answer: an autoclave is a device used to sterilize equipment and materials through the application of high-pressure steam. It is commonly used in laboratories, medical facilities, and industries that require the complete elimination of microorganisms, including bacteria, viruses, and spores. The autoclave works by creating a controlled environment of high temperature and pressure, which effectively kills or inactivates various forms of microbial life. The process is particularly important for ensuring the safety of medical instruments, laboratory equipment, and other items that come into contact with biological samples or need to be free of contaminants. Question 23. Labeled diagram of bacterial cell. Cell wall. Plasma membrane. Cytoplasm. Ribosome. Plasmid. Flagella. Pili. Capsule. Question 24. Laboratory diagnosis of tuberculosis. Answer: the laboratory diagnosis of tuberculosis, TB, involves a combination of techniques and tests that detect the presence of Mycobacterium tuberculosis, the bacterium responsible for TB infection. Accurate and timely diagnosis is crucial for effective treatment and control of the disease. Here are some key laboratory methods used for diagnosing TB. Microscopy, Ziehl-Neelsen staining. This is a basic technique used to visualize acid-fast bacilli, AFB, present in sputum, tissue samples, or other clinical specimens. The specimen is stained with a special dye, Ziehl-Neelsen stain, that makes AFB appear red against a blue background under a microscope. Microscopy is rapid and relatively inexpensive but has limitations in terms of sensitivity, especially in cases of low bacterial load. Culture. Culture of clinical specimens, usually sputum, on special media that support the growth of M. tuberculosis, allows for definitive identification and drug susceptibility testing. Culture is considered the gold standard for TB diagnosis as it provides accurate results and allows for drug sensitivity testing. Serological tests. Serological tests detect antibodies against M. tuberculosis in the blood. However, their accuracy and reliability are often questioned, and they are not recommended for routine TB diagnosis. Question 25. Universal precautions. Answer: universal precautions refers to the practice, in medicine, of avoiding contact with patients' bodily fluids, by means of the wearing of non-porous articles such as medical gloves, goggles, and face shields. Universal precautions are: use barrier protection at all times. Use gloves for protection when working with or around blood and body fluids. Change glove between patients. Use glasses, goggles, masks, shields, and waterproof gowns/aprons to protect face from splashes. Wash hands if contaminated and after removing gloves. Question 26. Name 3 viruses. Answer: influenza viruses, human herpes viruses, coronavirus, human papillomaviruses, enteroviruses. Question 27. Staphylococcus. Answer: Staphylococcus is a genus of bacteria that includes various species, some of which are common inhabitants of the human body while others can cause a wide range of infections. These bacteria are spherical, cocci, in shape and often appear in clusters resembling grapes under a microscope. Common species, Staphylococcus aureus is the most well-known and clinically significant species within the Staphylococcus genus. Other species include Staphylococcus epidermidis, Staphylococcus saprophyticus, and Staphylococcus hemolyticus, among others. Question 28. Organisms causing diarrhea. Diarrhea is a common condition characterized by frequent, loose, or watery bowel movements. It can be caused by various organisms, including bacteria, viruses, and parasites. Here are some of the major types of organisms that can cause diarrhea. Bacteria: Escherichia coli, E. coli. Salmonella, Campylobacter, Shigella, Vibrio cholerae. Viruses: rotavirus, norovirus, adenovirus. Parasites: Giardia lamblia, Cryptosporidiosis, Entamoeba histolytica. Question 29. Passive immunity. Passive immunity refers to the temporary transfer of preformed antibodies or immune cells from one individual to another, providing immediate protection against a specific pathogen or toxin. This type of immunity does not involve the recipient's immune system directly producing antibodies or memory cells but rather relies on the ready-made immune components obtained from another source. Passive immunity can occur naturally or be induced artificially. Natural passive immunity. Maternal antibodies. During pregnancy, antibodies are transferred from a pregnant person's bloodstream to their fetus through the placenta. This provides the newborn with some level of immunity to diseases the mother has been exposed to. Breast milk. Breast milk contains antibodies that are transferred from the mother to the infant, helping protect the newborn from infections until their own immune system matures. Artificial passive immunity. Antibody administration. In some cases, specific antibodies can be harvested from individuals who have recovered from a disease or have been immunized, and these antibodies can be directly administered to a person at risk of or currently suffering from the same disease. This approach is used in certain medical treatments, such as administering rabies immune globulin after a potential rabies exposure. Monoclonal antibodies. Monoclonal antibodies are artificially produced in the laboratory and can be used to provide passive immunity against specific pathogens or toxins. They are used in various medical treatments, including some cancer therapies and treatments for certain autoimmune diseases.